Welcome to General Biology. Today's video is going to be on active transport. Active transport can happen in a couple different ways. We can either move molecules across the membrane using pumps that run on ATP, or we can engulf molecules with the cell membrane and bring them in and get rid of stuff in the same way. So, if we're comparing diffusion and facilitated diffusion to active transport, a couple main differences. With passive transport, you're always moving from high concentration to low concentration. With active transport, you're always moving from a low concentration to a high concentration. So we usually use active transport when we want to store something. For example, I need to store sugar in my liver. So the sugar's in my bloodstream. I need to move it into my liver where there's already a high concentration of sugar. So I'm going to have to physically pump that sugar into my liver. This is a common protein pump that keeps our nerve messages constantly moving along our nerves. And basically what it does is it creates a gradient of sodium and potassium in our nerve cells. The sodium are constantly pumped out, and the potassiums are constantly pumped back in. And this creates what's called resting potential. Another type, other than the pump, is when we actually physically surround things and move them in, or use a vesicle to remove them. So we call those processes endo and exocytosis. So if we have a large molecule that we need to move into the cell, basically what happens is the cell membrane will engulf the molecule, and then the cell membrane will seal off here, and then there will be a vesicle with that molecule inside of it. Oh. So we have an example of it up here where the membrane pinches off and it forms a vesicle with that inside of it. Now what's going to happen is this lysosome is going to come up and bond to the vesicle, and it's going to break down the substances in here. The cell will absorb what it can use, and then the remainder will be released back out into the extracellular space. That process is called exocytosis. So endo to enter, exo to exit. So the easy way to remember that is EN starts endocytosis and enter, and EX starts exocytosis and exit. So here's the whole process in one big picture. We have a food particle that's taken in. The lysosomes come up and bind, dump their digestive enzymes. It doesn't show, but some of these molecules will move across the membrane into the cell. And then whatever's left over is going to be released into the extracellular space. This is another example of exocytosis. Every time a nerve impulse needs to move from one nerve to the next, there's a signal that happens that causes vesicles to bind with the cell membrane of the nerve, and they will dump their contents inside these vesicles as neurotransmitters, and they will dump their contents out into the synaptic space, and those neurotransmitters will bind to another molecule on the surface and create basically sodium channels will open, and when those sodium channels open, sodium can flow into the next and creates another nerve impulse. So in between our nerve cells, there's actually a chemical process that has to go on, and then it starts another electrical nerve impulse on the next cell to carry the message on further.